Greetings, guys, gals, and non-binary pals. Welcome back to the Amazing Plant Project. And the second installment of delving into the Biology of Plants textbook. Now, let me see if I can get this thing so it's closer. Alright. That's a little bit better. Okay, so hopefully the audio is picking up. It looks like the mic is on and working from that. The video seems to be doing okay, but nobody is currently in the chat, so not sure on that one. There is somebody watching though, so whoever you are, if you want to let me know if you can hear me, that'd be great. Right, so today we're doing the molecular composition of plant cells, which it's not the structure of the cell, but it's the components that go into making the structure of the cell possible, kind of. The actual physical structure is next week. So, things to look forward to. Let me just make sure that it's actually showing up on here, because I know last week when I tried getting the chat to work, it fritzed out on the computer and I had to use the phone, so. Okay, so I can hear the audio on here, so it seems to be doing okay. I'll just mute that to not have the backlog with that. And it seems to be lagging as usual. Okay. Right. Okay. Well, come on. Alright. So I've got that. Come on. What are you doing? Alright, so I've got that there. So kind of the same format as last time. We're gonna do like kind of the checkpoint overview, and then, or the checkpoints that the creators of the text are wanting you to know at the end of it. The overview, that's kind of the notes and stuff, terms and some conclusions and whatnot. Okay. This is just fritzing out again, so let's see if I can get this to work. Is it not working again? Seriously? Okay, 1 second. Just trying to restart it on the phone, so if it's my phone, then I know it's working for at least some of y'all. And if it's not, well, that's not good. So, in the meantime, hoping that it's just my phone being weird, because that tends to be a thing. I'm gonna go over the checkpoint intro bits, right? So, there's 7 questions that they're asking in here. So, 1, what are the 4 main types of organic molecules found in plant cells, and what are their basic structural subunits and their principal functions? Like, what What are the main four? What are they made up of? And what do they do? Seriously, it's not. Okay. I'm just going to restart this. And hopefully the rest of the stream is fine and dandy. Okay. So that's number one. Number two is by what processes are all four types of organic molecules, so the four that were discussed in the first one, what processes are all four types of the organic molecules split into their subunits, and by what process can these subunits be joined together? So what are the main four? What are their subunits? What do they do? And how do they join and split off in the process of everything happening? Yay! Hey guys! Good to see you in the chat. Alright. So, the third one. How do energy storage polysaccharides and structural polysaccharides differ from one another? This is why I edit my stuff. It's not really a thing if you're doing live. How do storage and structural polysaccharides differ from one another, and what are some examples of each? Number 4, what is an enzyme, and why are enzymes important to cells? Number 5, how is ATP different from ADP, and why is ATP important to cells? And number 6, what is the difference between primary and secondary metabolites? And 7, what are the main types of secondary metabolites, and what are some examples of each? So there's a range of topics that we've got happening up in here. So the real basic stuff, we're going to start with overview of exactly. Good on you for doing the homework. So yeah, ATP hydrolyzes to ADP, releasing energy. Very good. And that tells me that it's actually working still, which is awesome for the chat itself. Because when I tried getting it to show up on here, it just no, nothing. So, okay. Gonna dive into the initial stuff and then go from there. So, real basic level. Living organisms are made up of primarily 6 elements. So we've got carbon, which is where you get the idea of a carbon-based life form. It's just a living organism that has carbon as part of the cell makeup. So carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. The bulk of living organisms, shall we say, is water. So that's where you get the idea of water is life. And a few times in the text it talks about dry weight of something. That's basically like if you were to remove all the water from whatever the organism was, then that's what the remaining mass, constituents, what have you, would be weighted at. So, like, if something is half water and half other matter, then if it's like dry weight 50% protein or something, then it'd be like a quarter of the total mass, kind of. So, okay. So that part of it. And okay, yeah. So the bulk of living matter is water, and most of the rest of living matter, so the other, like if it's half water, then the other half, basically, is what we're talking about right now. Most of the rest of living matter is composed of organic or carbon containing molecules that can be broken up into carbohydrates, lipids, proteins, and nucleic acids. And we'll get into each of those in a little bit. So, okay, yeah. I was looking at the notes and circled the wrong thing, and it threw me off for a second. All right, we're good. So, there's, yeah. So, organic molecules in plants, just like in animals, really, but in plants are divided into four main categories. So, we've got the carbohydrates, the lipids, the proteins, and the nucleic acids. And the carbohydrates are kind of, they're made up of sugars and chains of sugars. They're the primary energy source energy storage. We'll say that. The sugars themselves are a primary energy source before it's processed, but the carbohydrates, because it's like a grouping of sugars, is a energy storage setup. So, have fun with the toasted carbohydrates. In most organisms. There's some that don't do that, but for most of them, yeah. And then you've got carbohydrates broken down into monosaccharides, which are the like simple single sugar molecules, like ribose, glucose, and fructose. That's pretty much what you see on most labels, packaging, that sort of thing. If you're getting some kind of snack food or breads or whatnot, that's the low run. And then you've got disaccharides that are two sugars that are covalently bonded. Like, sorry. And the return of I need water now. I know it's not going to pick it up on the webcam because I went back and looked at the stream footage and yeah. But the water bottle I've got has random plant stuff all over it from REI, which is pretty awesome and fitting a figure. So anyway, back to the topic at hand. Okay. Yeah, so disaccharides are comprised of two sugars that are covalently bonded. And for that, you have things like sucrose, maltose, and lactose, which are harder, shall we say, to break down. Admittedly, I hadn't heard of fructose being so much of an issue for people, but yeah, from what I'm aware of, at least, the lactose has the potential to be more of an issue, at least as a as a digestive thing, because of the structure. Because if it's a covalent bond, it's a stronger bond and it's harder to break down. So that would entirely make sense. And then we've got the polysaccharides that are many sugars, and that's where you get the cellulose and the starches. And going off that, yeah, the more complex the sugar structure is, the harder it is to break down, which there's a lot of organisms that can't break down cellulose or have a really difficult time breaking down cellulose. That's why, like, carnivores don't eat plant material unless, like, in the case of cats, for example, cats cannot digest plant material. But if a cat is feeling kind of like nauseous and just generally ugh, then they'll go out and gnaw on some grass or something because they're not feeling well. Sometimes it, lets them just kind of deal with whatever the issue is and it passes. Sometimes it's an irritant and it causes them to expel whatever the issue was that they were having, whether it's a hairball or they ate something funky or whatever else. So, yes, many sugars in the Twizzlers. Don't quite think that's what they were going for, but there are many molecules in the Twizzlers, so there we go. Yeah, for dogs, from what I've seen, it might be a little bit less, just because dogs seem to be more on the omnivore side of it. But yeah, it's the same principle for dogs too. But yeah, the cellulose makes up the walls of like the young plant cells, and is embedded in a matrix of other like carbohydrates and proteins that form the iconic structure of the plant cell walls and stuff, which we'll get into next week. But the really basic, like, animal cells are kind of circular, and plant cells are kind of squarish, rectangular, depending on where they are. The rigid cell structure is from the cellulose, and especially if you're vegetarian or vegan or otherwise mostly plant-based in your diet requirements, like moi, there's a reason why it's a good idea to cook a lot of things, because it breaks down the cellulose that you're working with and makes it easier to digest. Also why taking probiotics and certain yogurts and stuff is pretty good, because if you try and digest it just by itself, sometimes, depending on your own body chemistry and gut flora and stuff, and whatever the thing is that you're trying to digest, it's more or less difficult to process. And if it's hard to process for your body, then you're expending more energy to do the processing, and you might not get an actual return in the form of usable stuff. Yeah, having it saying that it's denatured by cooking, that'd work. It's the same principle as if you're chewing something, then it's starting the I hesitate to say the decomposition process because it usually infers like something kind of dead by the side of the road, but that's what's that's what's happening. There's the enzymes in your saliva are starting to break down whatever it is that you're chewing, which is, aside from not wanting to choke on whatever it is that you just ate, is a really good reason to make sure that you're chewing your food properly, because it starts the digestive process, so it makes it a lot easier. Yeah, exactly. It makes it a lot easier to further process it in the rest of your system when you're working with it. Because otherwise it gets to your stomach and it just kind of sits there sometimes, and that's when you start having a stomach ache. Unless there's some other extenuating circumstances that are happening, which, that never happens. Sarcasm. Okay. And then, right. Okay, so we've got the polysaccharides in there. And then there's macromolecules. Now, macromolecules, as the name kind of suggests, are large molecules. And like the previously mentioned polysaccharides, they're made up of identical subparts. And that's when you get things called polymers. So the subparts, or like the, shall we say, okay, yeah. Alright, and then the individual polymer subparts are called monomers. And polymerization is the process of linking the monomers to form the polymers. Molecular structure of sarcasm would be an interesting thing to create, though. Gotta put a pin in that one, because otherwise my brain will just, go off on a tangent, and we're in class time. Dang it. Alright, so let's say, the whole container of colored pencils here is the macromolecule. Alright. Then you've got the colored pencils themselves that are the polymers. And the leads in the pencil, so the lead and the wood, shall we say, are each monomers in the polymer that is in the macromolecule. So that helps to break it down at all. Yep, okay. Slight technical difficulty for a second there. Yes, all the visual analogies. Alright, so we've got that sorted. And now, so the four parts we've got. So carbohydrates, and the second one is lipids. So now we're on lipids. most lipids are fatty or are fatty contain fatty acids. And if you're looking at like oils and stuff, for example, that don't really mix with water, they don't dissolve in water, that's what we're talking about right now. So lipids tend to be in that hydrophobic or water fearing, that's the translation, water fearing, which means that they don't dissolve in water readily, sort of category. The primary purpose is another source of energy for the cells, as well as structural material. And although some are pretty large, the lipids can get pretty large, they aren't macromolecules because they aren't formed through the polymerization process. It works. The butter and the toast, you were saying about visual analogies, and they're tasty, so there you go. Okay. Okay. Okay, so there's a number of different components in the lipid category. So we've got the more well-known ones, perhaps, that are the fats and oils, the phospholipids, cutin, suberin, probably saying that wrong, but there we go, waxes, and steroids. And we'll get into those in a second. So, first we've got the energy storage ones that are the fats, oils, and phospholipids. So, get into that. So for the fats and oils, both are made up of fatty acids, and there's three main. What am I doing? Okay, yeah. And both the fats and oils are made up of three fatty acid molecules binded together to one glycerol molecule, and formed via the dehydration synthesis process, which is literally like the removal of H2O from the compounds. It's also sometimes called the can't remember right now, and it's bugging me. Condensation reaction. And they're pretty much the same, really. They just have a different kind of melting point, if you will. So generally fats are solid at room temperature and oils are liquid at room temperature. There's a few that are kind of in the mid-range of that. Like coconut oil tends to be solid until a certain heat point, and then it's liquid and stuff like that. But that tends to be the basis for those. And they are condensation reactions, present plants, somewhat for that. So with all the condensation reactions, surprising plants need watering in some ways. Somewhat, but also in order to take in the carbon dioxide they need, some of the moisture is released through the stoma from last week. So in that process, it kind of evens out and stuff, and if there's enough of a discrepancy between the interior and the external ecosystem, sort of, then there's some issues in the cell structure, so there's that. But yeah, if that's a built-in part of it, it's kind of interesting how stuff has developed over the millennia on that one. Alright. And the fats and oils are also triglycerides. So, and then we've got the phospholipids that are modified triglycerides that are usually part of the integral structure of the cell membranes, which okay. Okay. So phospholipids tend to where is it? Yeah. Phospholipids are composed of fatty acid molecules attached to a glycerol backbone. However, the third carbon of the glycerol molecule is linked to a phosphate group instead of a fatty acid, and there's a polar group that's attached to that. So because of that, instead of just being there, you end up having phospholipid layers forming kind of a bilayer sort of thing. So it would be nice to be able to actually have the images show up in the stream somewhere, because, like, I know it's not going to really work in that, but I'm going to try it anyway. So, there we go. If that's any kind of actually working. So, we've got so we've got the there we go. A phospholipid molecule consists of two fatty acid molecules linked to a glycerol molecule, as in a triglyceride, but the third carbon of the glycerol is linked to the phosphate group of a phosphate-containing molecule. The letter R denotes the atom or group of atoms that makes up the rest of the molecule. The phospholipid tail is nonpolar and uncharged and is therefore hydrophobic, so it's insoluble in water. The polar head containing the phosphate and R groups is hydrophilic, so it's soluble in water. So, you have the polar hydrosoluble hydrosoluble okay, one sec. Okay. So the hydrosoluble components are butted up against the water, and the tails that are not water soluble are in the interior. And with that sort of structure, because it ends up being like water on the two sides, the water soluble bits, and then the non-water soluble bits. And that ends up being a really strong component in the structure of plant cell membranes. Okay. And now we get to the components that are part of the forming barriers to prevent water loss. Okay. An intriguing idea. Admittedly, for that, I don't know. I have to think on that one. Alright. Okay. So, now we're on to the ones for forming barriers to prevent water loss. So that's the cutin, suberin, waxes, and steroids. So cutin and suberin both form a matrix that waxes are then kind of embedded into, and they're both important for the structural integrity in many plant cell walls. Also, okay. And then the waxes are long chain lipid compounds that kind of slow water dispersal, and the main well-known one, as far as plants are concerned at least, is the cuticle, which we discussed last week. But it's like the okay, yeah, there we go. So the cuticle is the layer that acts as kind of a protective layer, for lack of a better term, on the outside of the plant's epidermis to prevent water loss. And it's composed of wax embedded in the cutin. So it's cuticular wax or the cuticle. And there's usually a layer of epicuticular wax on top of that. Better depends on the environment. They still have, like, for succulents, they still have the stomata for respiration. They tend to have thicker cell walls. Succulents and cacti are one of the weird ones. Just because that's the only category, really, that if you try and dry specimens from, like, you're doing field work and stuff and you're drying the specimens, if you try and do that with succulents, it just resprouts in the drying chamber. For everything else, it just dries it. But for those, you actually have to douse it in alcohol in order to make a herbarium sample, which is an interesting idea, really. But as much as I like succulents, don't quite remember on that one, but I will make a note of it and get back to that. But it would make sense if, pretty much, yeah. I mean, ethnobotany is a fascinating subject, at least in part because of all the meandering things that you can do with plants, which we'll get into in a little bit too, with some of those. But for the wax, the last bit on that, one of the more well-known ones that has a number of uses is, I can never remember how to pronounce this, but carnauba wax. Carnauba. From the position of the A and the U, carnauba seems like it's right. Okay, we'll go with that. Carnauba wax that's used for polishing cars and floors and jelly beans, and various other sundry things that you need some kind of wax for. And that's from the Carnauba wax palm Copernica cerifera from Brazil. Yes, and boots and tic tacs too. Just polishes everything, pretty much. And okay, and then yeah, what am I doing? And then for the next category for that, we've got the steroids, which is also part of forming the barriers and preventing water loss. So, steroids have 4 interconnected hydrocarbon rings, and for that it's hydrocarbon chains and hydroxyl or hydrocarbonyl groups can be attached to these to make other structural molecules that help Yes, it is a Brazilian wax. But the okay. Okay. So for the steroids, right? And then when a hydroxyl group is attached at the carbon 3 position, the steroid is called a sterol. And I mean, it makes it more interesting, really. To be politically honest. It's good having actual, student people who are interested in what I'm trying to teach. So, it works. Alright, okay. Sterol, and then of the sterols available and whatnot, we've got alright. Admittedly, the chemistry part is not my strong suit. Yeah. Alright. So, cytosterol is the most abundant sterol found in green algae and plants, and ergosterol is frequently found in fungi. So, yeah. And cholesterol, which kind of fills the same niche of things is found often in animal cells. That it's only in certain trace amounts in plants, really, which is kind of fascinating. Wait, 1 sec. Computer, what are you doing? Just selecting random stuff. Okay. 1 sec. There we go. Okay. It's also interesting, too, because in pretty much every organism, aside from the prokaryotes, that's the single-celled, or rather, the cells that don't have a kind of discernible envelope around the different components in the cell. But the words, they have a pretty strong role in everything else, which is kind of cool. Alright, so we've gotten through the carbohydrates and the lipids and the carbohydrates and lipids. Okay, so now it's the proteins. I can kind of see that, yeah. I can see the steroids being considered as kind of a water wing sort of thing. Because, yeah, they're a stabilizing force. That's why they're applied, why they're used for medicines and stuff. Okay, so proteins. Proteins are the most abundant of the organic molecules, and most living organisms have at least like 50% protein dry weight. And again, we're going back to the idea of dry weight being like if all the water was removed, half of what's left would be protein. And amino acids are the building blocks of those proteins. Now, there's 20 different amino acids. There's a little bit of a plus or minus on that one. But there's 20 different amino acids broken up into 4 different categories. At that point, it's probably partially because of herbarium samples, partially yeah. Because I can tell you that a pound of fresh mugwort and a pound of dry mugwort are a completely different measurement. Mugwort's not too bad, but like there's some that there's a lot of what in there. And that would give you an idea of the weight of the actual plant structures, not the weight of the water in the plant structures. Okay, so Yes. Much experience on that one. Oh man, that's my nose is itchy. So, all right. So there's 20 amino acids broken up into 4 categories. The first one, we've got nonpolar hydrophobic. It does not dissolve in water. It doesn't want anything to do with water. It's just by itself doing its own thing. So we've got glycine, alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine. That's a word in and of itself right there. And tryptophan. Now, of that, tryptophan is probably the one that is most recognizable. Yeah. So, and for those, there's the general formula for an amino acid that is NH2, so 1 nitrogen, 2 hydrogen, and a carboxyl group that's COOH, so carbon, 2 oxygens, and a hydrogen bonded to essential atom carbon. Essential carbon atom. And there's also a hydrogen and a side group, so the rest of the atom, or the rest of the molecule, that's the R, that are bonded to the same carbon atom. And that's kind of the basis for all the rest of the amino acids. And whatever the R is, that's like, that determines what the rest of the process is that that particular molecule is going to be doing. So, got those. Then we've got polar uncharged hydrophilic. So, the ones that are polar but have no charge associated with them. No charge associated with them, and they are water soluble. Excuse me. So we've got serine, threonine, cysteine, tyrocysteine, asparagine, and glutamine. Polar negatively charged, that's also hydrophilic, is aspartate and glutamate. And that's what you might recognize from, say, monosodium glutamate. It's glutamate from that. And then we've got polar positively charged, that's also hydrophilic, so water soluble. We've got lysine, arginine, and histidine. Histine. There we go. So, those. All right. Okay. Just read the entire other side of the thing that I was going to go with, but okay. All right. So, I mean, tryptophan just kind of hanging around in an inner tube, just chilling. I mean, especially if they're in a body of water, that wouldn't be too far out of the realm of possibility. Just kind of milling around, doing their thing, and working on not being completely obnoxious. Yeah. Because, I mean, they do all have very particular jobs. Alright. Okay. Oh. Okay. Yeah. Alright. Okay. And we've got the nucleic acids over here. That's the fourth part. It consists of long chains of nucleotides, and there's two types of nucleotides. So we've got the ribonucleic acid and the deoxyribonucleic acid. So RNA and DNA. RNA has the ribose sugar subparts, and DNA has deoxyribose sugar subparts. Both are formed via dehydration synthesis reactions. So again, the breakdown of the water, the removal of the water. And there's alright, one second. Yep, okay. So within the sugar subunit bit of the nucleotides, we've got 5 different nitrogenous, we'll go with that, bases. That's basically the properties of a base plus a nitrogen. And that makes up the building blocks of nucleic acids, which make up the building blocks of DNA and RNA. So the building blocks of the genetic material that make up the rest of the cells and composition and stuff like that. It is a possible, but I unfortunately don't have experience eating the lab samples to best determine whether DNA or RNA has a sweeter flavor on that one. Of course, RNA tends to be the simpler structure and like and stuff like that. And DNA tends to be pretty much everything else, or RNA operating within the capacity of making the DNA do what it's supposed to do. So we shall see on that one. But yeah, so nucleic acids. We've got adenine. That, where did it go? No. It's not right. It's not written down because it's not in there. Got it. Alright, so adenine, guanine, thiamine, cytosine, and uracil. Now, that right there, those 5 are the really important parts. And we will get back to that at a later date. But if you look at a DNA sequence and see like AGTCU in whatever order, that's what it's referring to. The first letter of the compound is uracil. Let me see where it actually comes in again. Yeah. Pyrimidine primarily found in RNA but not DNA. Or found in RNA but not DNA. Kind of. And the same with thiamine. So uracil and thiamine are both in RNA but not DNA. What's Gattaca in this context here? Okay. And so we've got DNA that is the carrier of genetic messages. So the genes, the things that tell the cells and the components of cells based on the genetic data what they're supposed to be doing. Then we've got RNA, that's molecules that are involved in the synthesis of proteins based on the genetic data provided by the DNA. So you've got the DNA that has the genetic memory, and then you've got the RNA that kind of follows whatever instructions the DNA sends out. Okay. Because that's the only context that I've heard the term Gattaca. So just making sure it wasn't something else that I wasn't aware of. But yeah, sci-fi. Okay. I didn't know that DNA was that important a plot device in there, though. Interesting. Very cool. Okay, so that's the DNA and RNA. It will be revisited rather a lot, probably. And if it's not via the text, it will be brought up again because it's fascinating. So, past that, we've got we've got a water break, apparently. Alright. So, we've got adenosine triphosphate, or ATP, and adenosine diphosphate, or ADP, which I have found my whiteboards, so I don't know if it's actually transmitting properly or if it's reversed on here, but let's check with the thing on the phone and see. Come on. Aha! It's working. Fabulous. Okay. So, we've got the ATP that becomes ADP through the process of hydrolysis, and then you've got ADP that becomes ATP. Jeez. Once you add in the phosphate and oxidization. So that is the process on that one. So ATP is the principal energy carrier for most processes in living organisms, and ADP is the products of hydrolysis. Well, the products of hydrolysis are ADP, free-roaming phosphate, and energy that can be used by the plant or the animal or other creature that consumes the plant. I mean, yeah. Any kind of plant-related science would also be prime real estate for going up on the channel too, because yeah, if there's any particular shows or general pop culture things or whatnot that would be of interest to see dissected from a botanical perspective, let me know and I'll look into it. I have a whole list of manga and anime for that, and a few other shows and books and stuff. So yeah, it's an ongoing thing. I'm just trying to figure if like Western originated graphic novels like The Last Unicorn that I have over here and a few other ones could be incorporated under the ethnobotanime label for like posting and stuff. So if you have any ideas, let me know. But yeah, Gattaca sounds like that'd be pretty cool. I haven't seen that, so yeah, very nice. During the process of respiration, ADP is recharged to ATP when glucose is oxidized to carbon dioxide and water. Actually, because I was reading it. This one. And it's super awesome because it's like fully illustrated and everything, and it's like it's super cool. Oh wait, one sec. Trying to put it back where it was, and there's stuff falling over. Okay, we're good. Yes. Oh. There we go. Okay. So there is that. So ATP is the molecule directly involved in providing energy for much of the cell as it is alive. So, yeah. Like, I've got the regular camera angle over here, and I've got the phone over here, so I can just make sure that everything's still working. And it's throwing me off because that one is like what you're seeing, and this is mirrored, and it's like so for it ended up being a lot more convoluted than I wanted it to be when I drew it out, but we've got the cycle process kind of. So we've got the three the three phosphates up here, and and ribose. And then you've got adenine attached to it. And then you add hydrogen, and it becomes, so this is ATP up here. And then through and then via hydrolysis, we've got the 2-phosphorus attached to ribose and adenine, and then the free-ranging phosphorus and energy over here. So that just reinforces that I can in fact use whiteboards and have it work, which is cool, because I can even see it on the tiny screen on the phone. So fabulous. Much good. So nice. Awesome. So, got that. And we're moving on. So, now we've got the sector of alkaloids. Now, admittedly, chemistry wasn't particularly my main strong suit. However, alkaloids are rather fascinating. If you have not listened to it as yet, I have a podcast entirely devoted to the darker, sort of creepier side of botany. Now, sometimes that there's world events that have issues and things, like the mass fires in the Amazon last year. There's a few posts there regarding that. There are some that are like climate change and things. But a lot of them, most of them, are regarding the poisons, potions, and brews, shall we say, that come out of the natural world. So, toxins and things in tomatoes was one of them. And then there's other things like that. There's a lot of them. For those, I'm really selling it, I don't know. But, I've not posted a lot on that recently, and recently being like the last couple of months. There's going to be more going up in the next few weeks, and most are going to be things like this, where there's mention of the alkaloid producing species. So, we've got, and I realize it's probably not going to show up too well, but there we go. So there's these guys right now. So, the ones mentioned in the text. Yeah, morning glories are pretty cool. And there's some interesting euphemisms with those, so yeah, stay tuned for that. But the ones mentioned in the text, as it were, are cocaine, which in its natural state is just in the coca leaves. So erythroxylum, erythroxylum coca, which it gives kind of a high, sort of, not a high, but like it's kind of an appetite suppressant and helps you sort of continue working for an extended period of time, especially for that. For caffeine. There's caffeine in chocolate. And for the coca or cocaine, it's native to the Andes Mountains in Bolivia and Peru, and then the locals tend to just kind of chew some of the leaves. It reduces hunger pains and fatigue while working in the kind of hostile environment that exists there. The problem comes when it's synthesized and turned into cocaine as it's known in general. When it's like the crack cocaine format, if it's smoked and like injected and everything, or if it's used in excess, just like for pretty much anything. If it's done to excess, then there's a problem. If it's not, then it depends on the individual. Second one, we've got caffeine. Now, that's probably the most acceptable drug as such. We've got coffee, tea, and chocolate, or cacao. Now, depending on the variety of coffee and tea and stuff, there's more or less caffeine sometimes, depending on the environment that it's grown in and different conditions that are happening. Just like for anything else, the makeup and expression of certain compounds vary depending on the environment that it is from. That's one of the reasons that, well, I fully appreciate like naturopathic medicine and like taking your health into your own hands and doing what you need to do for yourself with that. You have to make sure that whatever compounds you need from the thing are present. Because sometimes you might need, like, let's say there's 10 milligrams. You might need 10 milligrams of a compound in whatever the substance is. And plant X has 5 and plant Y has 15. Wait, seriously? 1 second. People are snorting cocoa instead of cocaine? Really? That's a thing? Really? I want to say that I'm surprised, but then I'm not really. People, why? Just why? Of course, people were also like trying to eat like half a bottle of nutmeg a while back too, so I'm not entirely surprised at that point. But like, oh man. And the nutmeg one doesn't even make sense. Like, you get kind of high, but you get really nauseous first, and it's just every report that people have had where they've done that and come down from it, it's just a really bad trip. So, like, why would you put yourself through that in the first place? It makes no sense. It's not even a good experience. Like, that point is just idiot bragging rights. Like, nobody needs that. Why? Come on, man. Ah! I don't get it. I really don't. Alright. So, back to that. We've got, so, the ones mentioned in here, we've got cocaine, caffeine, nicotine, and atropine. So, nicotine is the second most okay, I suppose. One. Okay, there we go. from the tobacco plant. Now, for that one, it's interesting because oh yeah, forgot that. But for this part, yeah, socially acceptable. Well, there's less stigma around smoking tobacco than there is about smoking other things, depending on where you are. That's all. Things like cannabis tends to be tobacco, cannabis, and then like everything else is down here somewhere. Again, it depends on where you are. Some places it's like not at all. Some places it's whatever. So there you go. But it's interesting because for nicotine, it's synthesized in the roots and transported up to the rest of the plant. And it's created as a response to the plant having some kind of injury that it needs to fight off. Because mostly nicotine is created by the plant to deter grazing by herbivores and insects. As to why people don't smoke the roots, it's kind of a twofold. Partly, it's produced in the roots and then is sent to the leaves, and it's stored in the leaves. So if you want something that has a high concentration, it's the leaves. From a practical economic standpoint, if you try and harvest the roots, then you would have to uproot the plant in order to create more of the product. Whereas if it's the leaves, then you can just harvest the leaves and let it keep producing until that plant is expended. All right. Then we've got, and then we've got atropine that is found in things like Egyptian henbane that's I can pronounce it in my head, but actually vocalizing the Latin is difficult sometimes, so just bear with me a sec. All right. Hoyosacamus and Hoyosacamus muticus that was used by Cleopatra in the 1st century BCE to make her eyes more like alluring. Yeah, it's more of a, like, it's produced and sent off in the roots. It's, yeah. And that's the same principle for how deadly nightshade or Atropa belladonna was used in Europe for most of the medieval period. It was used Okay, one sec. Okay. Okay, staring. I think I ended up deleting your last comment, unfortunately. So I can see it, but it's not letting me reinstate it, which is obnoxious. Okay. But yeah, killing women for beauty since BC, which, accurate, really. Ah, there we go. There we go. Hit the wrong button. Okay. Alright. But Trump belladonna. And I can't remember if I've actually done an episode on the podcast about belladonna or if I've just thought about it. I think I've just thought about it. But there's a lot of different nightshades because that's a category of plant. Tomatoes are a nightshade. There's other ones that are a nightshade, but deadly nightshade isn't like it's got toxic components in it. Yeah, okay. Good to know. It's got toxic components in it, but it's not a deadly one per se unless you eat a number of the berries or if you miss ID the plant or if, like, say, a small animal or a child gets a hold of it. The issue for most of this is when you're looking at the old literature, and I have a copy of Gerard's Herbal that has the old names and everything before it was using the binomial nomenclature. And in that, there wasn't really a standard spelling for most of the species, and B, common names were used instead of or synonymous with the scientific names that were being used at the time. And one of the things that I found for that is, especially for the nightshades, because it's from Europe and there's a lot of different species native to Europe, you have the deadly nightshade and you have the woody nightshade. And woody nightshade is the more robust one, hence woody. And that one tends to have the more problematic toxins versus the deadly nightshade that is usually like confused with the woody nightshade. So you don't want to eat either of them, really, but I can say from experience, I've had some of the deadly nightshade berries, and they just taste like tomatoes. As long as you get them when they're fully ripe, and you're not trying to eat the green fruits, it's just like the black, dark purple ones. That being said, there's a lot of people that have reactions to those species. So, yeah. Also, if you don't know for certain what the plant is, I would not suggest trying to eat it. I only did that because it was growing in the garden of a friend who knew definitively that that plant was fine, and her chickens had come up to it and eaten a lot of it and seemed to really like it, and they were fine. So be aware of that. But yeah, Atropa belladonna on that one. And then we've got morphine. That's the final of the 5 going in here. Now, morphine is a byproduct of the opium poppy. And I've got the startup of a podcast post on the opium poppy that is probably, if I can finish the script, it's going to be going up this week or next week. But it was the first alkaloid actually identified in 1806, and oh, okay. That's the first that I've heard of the King's Gold. That's cool. And so yeah, the opium poppy is Papaver somnifera. One of those times that I know how to say it, but like the brain and the vocal doesn't quite compute with that. Okay, here we go. Papaver somniferum. There we go. And it's one of the more prevalent analgesics or pain relievers currently, but it also has the potential for a very strong dependency and potential for addiction if it's overused. Now, just like cocaine has potentially good properties, or coca, depending on how it's used and modulated, the morphine or the opium poppy tends to be used for pain relief and suppressing coughs. And there's almost 10,000 different alkaloids, like in the botanical sphere, just kind of naturally existing. But yeah. Alright. So, we've got the alkaloids out of the way. And that was a fun discussion though. And now we've got the terpenes. Now, terpenes or terpenoids, terpenin, that was an extra N in there, terpenoids, occur in all plants and are the largest class of secondary metabolites, which the primary metabolites are molecules that are found in plant cells and are necessary for the life of the plant. That's like the amino acids and nucleic acids and proteins and things like that, and sugars. The secondary metabolites are found in some plants and generally some parts of some plants. And many serve as chemical signals that enable the plant to respond to environmental cues and activate when there's some kind of threat that needs to be addressed, whether it's a herbivore trying to eat the plant or some kind of bacterial or fungal infection that's trying to come in. And there's other ones that aid with pollen and seed dispersal or protection from radiation against the sun, because there are plants that have light sensitivities that need assistance from their internal components. And like that, there's some that are termed phytoalexins that are antimicrobial compounds that are produced only after the plant has been wounded in some way, after an attack by a bacteria or fungi or something like that. And those can often vary during a 24-hour period too, depending on the necessities for that particular plant or species or what have you. So the terpenoids are the secondary, or the a little bit, yeah. All right, so the terpenoids are the largest class of these secondary metabolites, and there's over 22,000 different compounds that have been described or identified. Because scientifically, if it's something that has been described, then it has been identified and analyzed so that the scientist people involved can discern that whatever it is a particular sort of thing. It has been given a description, so therefore it has been described. The simplest of these is the hydrocarbon isoprene that's C5H8, so 5 carbons, 8 hydrogens. And all terpenoids can be classified according to their number of the isoprene units. So that's the main building block. That's the base Lego that they're working with. And everything else is 5 of those or 10 of those or what have you. I mean, that works. But, yeah, I mean, fungi would work. Also, anything that has an exoskeleton, which most insects, let's be real. But yeah. All kinds of fun things. Wait. Did I go past that already? I did. Okay, yeah. So, slight backtrack. Completely forgotten about the chitin. Chitin? Chitin, yes. Chitin. And it's a pretty important structure of the polysaccharides from, an hour ago. It's a principal component in fungal cell walls, and it's also part of like hard, dense outer coverings known as exoskeletons. So things like lobsters and crabs and the roly-poly pill bugs and pretty much anything that has an exoskeleton has exoskeletons of insects and crustaceans and things of that nature. But it's fascinating that something like a lobster and something like a mushroom have the same kind of it is chitin. It is. Okay. Yeah. Okay. See you later, staring. But yeah, it is chitin. I'd forgotten about that. That's why, if you're looking at ancient, if you're looking at archaeological sites, a lot of times, especially if it's coastal areas, and at this point I'm referring to California coastal archaeosites, there's a lot of deposits for plant material. Well, no, not plant material. Like sea-dwelling creature material. A lot of that is either fish bones or chitin. At least that's the terminology that was used in my field schools. Now, chitin was referring to a large pillbug type creature that was also kind of water dwelling, that had large like plates along the back like pillbugs do. And they're still around. I just haven't seen them for a while. But that sort of critter was termed chitin, which is pretty cool because that's what the makeup is called, or that's what it's made up of in the molecular composition. So that's pretty awesome. Okay. And it's a lot easier to say than chitin, too. That sounds better. So we've got the chitin on there. Then isoprene itself is a gas emitted in fairly significant quantities from the leaves that's responsible for the kind of bluish haze that you see on a hot day above and around a forest sort of area. And it kind of just hovers over wooded hills and mountains in the summer. And there's theories that that's because it's aiding the plant in dealing with the heat by stabilizing the photosynthetic membranes in the plant cells. And that's kind of further supported by the fact that it only happens during the day or during like in the light, and is made in the chloroplast from carbon dioxide recently converted to organic compounds by photosynthesis, which is pretty cool. Next, we've got essential oils. Now, essential oils are highly volatile compounds. And they're generally contributing to the fragrance of a particular plant or the essence. And that's why they're called essential oils, because they're a contributing component to the essence of the plant that they are part of. And a lot of these are either monoterpenoids, that it's just one terpenoid, or sequesterpenoids, that are 3 terpenoids together. And one of these, or the mint or mentha, has a large quantity of the volatile monoterpenoids, the singular terpenoid ones, of menthol and methone, that are both synthesized and stored in glandular hairs known as trichomes, that are outgrowths of the epidermis. Now, trichomes are not unique to mint plants and stuff like that. They're famously part of cannabis plants. That's a big part of it. And other species like that. And the essential oils in certain species, like some are meant to attract pollinators, some are meant to keep herbivores away so they're not eaten, some are meant to do any number of things, or like attack bacteria and fungi. There's some, like the diterpenoid taxol, so a 2-terpenoid compound that has anti-cancer properties and has been shown to shrink ovarian and breast cancer sometimes in certain circumstances. And the usual source for that has been the Pacific yew tree. Now, the issue with that is that even if you harvested all the bark from the yew, which is where the concentration is from it, you would still only get a small amount, and it would kill the tree, because the tree needs the bark in order to continue functioning. And it's interesting because they've done studies. Now, yew tends to be pretty volatile and poisonous. However, in certain situations, in certain concentrations, just like with the belladonna and foxglove and other volatile poisonous compounds, sometimes it can be beneficial. So in this instance, they've taken the needles from the European yew tree and figured out how to get a Taxol-like compound from it that has similar outcomes. They've tried doing a synthesis, like a lab synthetic version, and it hasn't quite worked. But, you know, there we go. Still in process on that one. And actually the largest known terpenoid compound currently is rubber. Now, there's when it says the largest, it's molecules containing 400 to more than 100,000 isoprene units. And that the isoprene being the C5H8 that we talked about before. Now, when it's obtained from the tree, it's a milky sort of fluid called latex. And most of it is from the Havia brasiliensis plant, generally called a rubber tree because it produces rubber. Now, there's about 1,800 different species that have been reported to have some kind of rubber component, but most of them what's that? But most of them don't have enough to be commercially viable as a source. Yeah. And then a lot of terpenoids are poisonous even. So the main, or the, and part of those are the cardiac glycosides, which foxglove or digitalis plants tend to be one of the more frequently cited ones, like that have the most active cardiac glycosides and digitoxins, which help to kind of it's one of those that if you need it, it works really well. If you don't, then it gives you an issue. But also, milkweed and dogbane are also poisonous and have particular toxins that specifically target herbivores to try and not get them eaten, as it were. However, there are certain species like the monarch butterfly that has specifically adapted so that milkweed is its food and habitat of choice. And in many instances, if the milkweed population is in decline in an area, so is the monarch population, because that's that's their food source. That's where they are. And for the monarchs, they've adapted so that they ingest the cardiac glycosides and just kind of store them as a defense mechanism. Because then if a bird tries to eat one of the monarchs, then it makes them really nauseous and sick, and they end up vomiting it back up. And most bird species have learned that this thing that looks like X, whether it's the caterpillars that are yellow with black and white stripes, or the butterflies that have the brilliant orange yellow spread with black veining, are poisonous, which is also why you see mimics that have that same sort of color palette but aren't monarchs. The same with flies and other smaller insects that look like bees, because predator animals have learned that bees are not a good food source because they sting and they hurt. And so if you look like the scary, threatening thing, then you're less likely to get eaten. Okay. Alright, so we're almost to the end for this. Just got a little bit left. So we've got phenolics, that is no. So phenolics encompasses a broad range of compounds, all of which have a hydroxyl group, that's the OH, attached to an aromatic ring. So a ring of 6 carbons containing 3 double bonds. And they're almost universally present in plants that are known to accumulate and are known to accumulate in all parts of the plant, like the roots, stems, leaves, flowers, and fruits. Now, they're the most studied secondary metabolites, but the actual function is still relatively unknown, which is an interesting sort of thing. So even with all the analysis, they're still kind of a mystery. Okay. Then we've got flavonoids, that are water-soluble pigments present in the vacuoles of plant cells and represent the largest group of plant phenolic compounds. Now, they're generally found in red wines and grapes and grape juice and stuff, and have had a lot of attention recently for the reported benefits for lowering cholesterol in blood. Now, there's over 3,000 different flavonoids that have been described, and it's probably the most intensely studied sector of phenolics, and it's divided into different classes, including the widespread anthocyanins, flavins, and flavonols. Now, the anthocyanins range in color from like red through purple to blue. And most of the flavones and flavonols are kind of yellowish or ivory colored pigments, but some are colorless. And the ones that are colorless tend to alter the color of the plant part through formation of complexes with the anthocyanins and metal ions called copigmentation. And that's usually what's responsible for the really intensely blue pigments in certain flora. And flavonoids also affect how plants interact with other organisms, like the symbiotic bacteria living in plant roots, as well as microbial pathogens and stuff like that. They also provide protection against damage from UV radiation. We've got tannins also. So tannins are the phenolic compounds present in relatively high concentrations in the leaves of a wide variety of woody plants. Now, a lot of times you see those in like the oaks and things like that. Like if you're, I know for inks, if you're wanting to make your own inks or your own dyes, a lot of times you go looking for oak galls. So most of the time for that, it's a gall wasp has kind of formed a growth on the underside of an oak leaf. Sometimes there's a whole range of them on a particular tree. Sometimes there's multiple species represented. Sometimes it's just like one or two. But the oak gall itself is the wasp's kind of nursery space. And that gall has a high concentration of tannins, just based on where it is and the interactions from the enzymes of the wasp and everything else. And that's a really strong dye if you're doing natural dyeing or you're making your own inks and things like that. Yeah. And tannins are sequestered in vacuoles within the plant cell in order to protect the other cellular components. And yeah, it's used to tan leather and denature the protein of the leather and protect it from bacterial attack. So it's pretty dang cool for the leather stuff. And we've got lignins that, unlike phenolic compounds, are deposited in the cell rather than in the vacuole. And it's second only to cellulose as the most abundant organic compound on Earth. And their polymers formed from 3 types of monomers, including p-coumarol, coniferol, and synephyl alcohols. the amount of each monomer kind of differs depending on whether the lignin is from a gymnosperm, so non-flowering seed plants, or woody angiosperms, so the woody flowering plants, or grasses. And there's a lot of variation with that too. And then we've got lignification, that's the process of lignin deposition that's believed to have played a major role in the evolution of terrestrial plants. Because for that, you're depositing the structural lignins, the structural supports into the cell walls to kind of secure them better, which it's kind of the same principle of trying to stack a pile of bean bags on top of each other versus a pile of boxes, or a pile of boxes that have sturdy cardboard and are reinforced versus a pile of boxes that are made out of paper. And yeah, with the addition of the lignin in the cell walls, it was possible for terrestrial plants to increase their height and develop branching systems capable of supporting large photosynthetic surfaces, like the large spanning tree limbs and the canopy of leaves. It also waterproofs the cell walls, allowing the cells to just kind of retain their moisture and oh man. And further supporting the upward transmission of that water. Because if the cells just kind of keep losing the water that they're holding, it can only go so far. But if they're able to keep it where it needs to be and push it up, then it can get up to the canopy too. Oh man. And then there's also wound lignin that protects the plant from fungal attacks by increasing resistance of the walls to mechanical penetration, so like cutting, and insects and birds and things trying to peck holes into it. And then protecting against fungal enzyme activity, because that's how the fungus would send out itself into a space that it's trying to invade via the enzymes. And it reduces the diffusion of enzymes and toxins from the fungi into the plant, which is pretty cool. And because it does that, it's been suggested that lignin may have initially functioned as a kind of antifungal, antimicrobial agent that only later took on the role of a water retention transport compound. Right. Then we've got salicylic acid, that is an active ingredient in aspirin and is known for its analgesic, pain relieving properties, which was discovered by both the ancient Greeks and the Native Americans It's from the willow bark, so salix, and it's only been recently that the phenolic acid in the plant tissues that has been studied has been discovered, and it's essential for the development of systemic acquired resistance, commonly referred to as SAR or SAR. SAR develops in response to a localized attack by pathogenic bacteria, fungi, or viruses. As a result, other portions of the plant are provided long-lasting protection against the same and unrelated pathogens. So, essentially, salicylic acid is operating as a vaccine, kind of, which is pretty cool. In the context of the plant itself. When it's removed in aspirin, then it's just aspirin, and it makes you feel better if you're not feeling good. But in the context of the plant, in certain situations, that's what has been studied as of the publication of the text, at least, which is pretty cool. Yeah, and it's for that, just like with anything else, it's important to remember that organic molecules are all kind of important and studied in their own right. They're very rarely functioning in isolation, and it's important to remember and study the interactions between them, because if part of the equation goes awry, if part of it doesn't work, or it's just not applicable for whatever reason, then you still need to see how the rest of it functions together, which is pretty cool. There was another. Okay. Yeah. When we were talking about caffeine, I forgot this a little bit. There are certain plants that will send out signals that are designed to inhibit or promote growth of other plants. Coffee happens to be one of them. Or, well, caffeine possessing plants happen to be one category of that. So, yeah, especially coffee though. So, which is part of the issue with coffee plantations, admittedly, because even when they've tried to grow undercover or undergrowth sort of plants, or other things in the general vicinity, the coffee plants themselves send out toxins. So that's something to keep in mind too. But the high concentrations of caffeine in the developing seedlings of the coffee plant have been shown to be highly toxic and lethal to both insects and fungi. In addition, caffeine released by the seedling apparently inhibits germination of other seeds in the vicinity of the seedling. preventing the growth of competitors. This process is called allelopathy. So, there is that. And yeah, thus concludes chapter 2 of the text. So, alright. So, I know Staring was the one in the chat. If anyone has questions that y'all want to ask, now would be a good time to do it. Otherwise, that's about it for today. Let me see. Okay. Instead of a quiz right now, how about you leave a comment in the comments section after this is over, with your favorite, either favorite because it's interesting or favorite because it's just something that you're really fascinated by or you have a question about, whatever, compound or problematic / misunderstood plant. So, until then, so that being said, I will leave you to what you're doing for right now and see y'all next time. There should be a new video up in the next couple of days, and then the next episode of the class time is going to be, again, next Thursday at 1, generally like 1 to 3, 1 to 3:30-ish. And for that, we're going to be going over the plant cell and the cell cycle. And that's when we're getting into the actual cell makeup. Again, if you want to get your own copy of the book or anything, there's links and everything in the description, along with stuff for the podcast and the blog and everything else. And my other channel where I'm doing more art type stuff. So until then, I will see you next time. Have a fabulous day and hope you're doing all right and having fun with the plant stuff. So, till next time, see y'all later. Bye, everybody.